Welcome to Basic Brewing Radio for Thursday, April 28th, 2016. I'm James Spencer. Here at Basic Brewing Radio, we're all about homebrewing. This week, we head back down to Nelson, New Zealand for Dr. Chris White's keynote presentation on yeast. What else? Chris presented some very interesting behind-the-scenes info about White Labs and some practical advice for homebrewers. If you're new to homebrewing or would like to get into the hobby for the first time, check out our website, basicbrewing.com, where you can find archives of our audio and video podcasts and our DVDs to walk you through basic and more advanced brewing techniques. And if you buy any of our DVD combos, you get a free Basic Brewing bottle opener. And don't forget to get a copy of our Brewer's Logbook. You can use it to log and track up to 50 batches of beer. You can follow me on Twitter. My username is Basic Brewing, all one word. We also have a Basic Brewing Radio and Basic Brewing Video page on Facebook at facebook.com/basicbrewing. Thanks again to everybody clicking on the Amazon.com associate link on our basicbrewing.com site. Whenever you think of Amazon shopping, think of us first and go to basicbrewing.com, click on our associate ad. It'll take you back to Amazon where you can shop as you normally do. You won't notice any difference. It won't cost you any extra, but you'll be helping us to bring you the show, and we greatly appreciate your support. We also have associate links for Brew Your Own magazine and the American Homebrewers Association on our site as well. You can find our Basic Brewing iPhone app on iTunes, our Android app on Amazon.com. We have a Windows Phone app. We're on the BlackBerry Podcast Directory, and we're on the Stitcher app as well. And I'm working on the Google Play thing, so stay tuned for that. And appreciate everybody letting me know that that's a thing. So that's another thing that I've got to do. If you want to put a tip in our tip jar, some coinage in our guitar case, you can go to basicbrewing.com/support. And thanks to everybody who's done so already. And if you do, I will send you some links to some bonus stuff that Steve and I have put together over the past 3 years. I got some fun news from New Zealand this past weekend. It's a New Zealand-themed show again this week. The Homebrew Shop, owned by Mike Stringer and Ed Bream, organizers of the conference down there, two of the organizers, that shop is called League of Brewers. They had a first anniversary get-together this past weekend, and they served the 5 beers that we Americans brewed at Marchfest on stage. You may remember that show where I feature that. Andy Sparks, Chris Colby, John Palmer, and Tasty McDowell and I all brewed beers on the Grainfather system in front of that crowd. I was worried about my cream ale recipe because the flaked maize that we used was less like flakes and more like powder. It was kind of like cornmeal. So when we stirred it in the mash got stuck. But thanks to the resourcefulness of my brewing partner, Carl Summerfield, we muddled through and collected enough wort and brewed the beer. And, boy, I can't tell you how much pressure that I felt, going up against those remarkable brewers there on the stage with me. My recipe against those other guys, forget about it. So I just hope that my beer was drinkable. So fast forward to this past weekend, the League of Brewers posted on Facebook that they were serving all 5 beers on tap at their anniversary party, and they said the first to run dry would be the winner. Well, I didn't know there was going to be a contest. Well, imagine my surprise when a later post showed that the beer that Carl and I brewed came out to be the favorite. I guess it ran out first. So I tried to give all the credit to Carl, but Carl wrote me and said it was a delicious beer. I think it was squarely a combined brewing effort, and it was your recipe. The most credit I am prepared to accept is 37%. So I guess I'll take 63% credit. Boy, I'm just shocked. The Kiwis are planning to send some bottles of each beer up this way, so I'm hoping that happens, and I'm looking forward to tasting them side by side to see if our beer actually measured up or if our keg had a leak in it. Thanks again. Thanks again to Carl for a stellar job in brewing that beer. And by the way, here is the recipe for the Riwaka Cream Ale. Get your metric hat on. It's a 19-liter batch. We used 4 kilograms of pale ale malt, You can use American Turo. 500 grams of flaked maize. We used a single rest mash at 65 degrees C for 1 hour. We used 55 grams of Styrian Goldings pellets at 60 minutes, 55 grams of Riwaka pellets at flame out. I'm really excited to taste what the Riwaka pellets tasted like. And California ale yeast. The original gravity was 1048, the final gravity was 1010, and the ABV was 5%. Now that's what I predicted with the recipe. I'm not sure what the actual figures were. And that recipe is also posted on our Facebook page. Now, Steve and I are planning to get together this week to shoot an episode of Basic Brewing Video, and I want to feature the latest incarnation of my rye wit that I brewed with some Nelson Sauvin hops from New Zealand that I brewed before heading down south. Over time, it has become very clear and tasty at 2.5% ABV, so it's very sessionable. Steve has also brewed a session beer, an ESB. It's the first time he's brewed on his electric brew in a bag system from our sponsors, High Gravity in Tulsa. Now, I have the larger 240-volt system that can brew from 4 to 10 gallons, and Steve now has the 120-volt system that can brew from 3 to 5 gallons. Both have the metal colander inside where you hang your bag, and both have the EBCSV electronic controller with a pump that you can use to keep your mash at a rock-steady mash temperature. Both store easily because everything that comes with the system fits inside the brew kettle, so it's got a compact footprint for storage. The difference comes with power. Mine can raise the wort or water temperature at a rate of 4 degrees per minute, while Steve's is at 2 degrees per minute. However, Steve's is more compact and you can fit it on the stove, so if you're in a hurry, you can use your kitchen stove to help boost the process and help reach your strike temperature or boil faster. Both are awesome. It just depends on what you want to do. You can check out a video that Steve and I produced to show off the two systems at youtube.com/highgravitybrew, or you can see all the details at High Gravity's website. That's highgravitybrew.com. And thank Desiree and Dave of High Gravity for continuing to support the podcast. highgravitybrew.com. Okay, Dr. Chris White, founder of White Labs Yeast, gave a very interesting keynote at the New Zealand Homebrewers Conference, and during that talk, he covered their tasting rooms and the new pure pitch packaging. Well, since we covered those areas very well, I thought, in our one-on-one conversation with Chris in the July 23rd, 2015 Basic Brewing Radio episode, I edited those sections out of this week's talk. But if you haven't heard it, please go back to the July 23rd episode from this past year. The White Labs tasting room is incredible, and the story of how pure pitch packaging came about is very cool. What remains of this keynote is some good science talk about yeast and some good advice on brewing with yeast, including some frank advice that is making me rethink how I aerate my wort.
Dr. Chris White of White Labs, the man who literally wrote the book on yeast.
Hi, everyone. We're just getting the computer set up here, but thank you for a great reception here in Nelson. I've been here for a few days, and hospitality has been awesome. Big program, big attendance, so that's awesome. Really happy to be here. So I'm going to tell you a little bit about yeast, right? We want to all make better beer, and yeast is a big component of beer. So I'm going to talk about that, but I'm also going to talk a little bit about White Labs. It's my first time here to introduce you to that, and San Diego a little bit. So for those of you who have been there. So here's a little White Labs here based in San Diego. Sometimes people ask me why I started White Labs in San Diego, and It wasn't anything really special except I was living there. I went there for graduate school after UC Davis. So that gave me some background in fermentation science at Davis. It has one of the few fermentation science programs in the United States. And I just was fortunate enough to go there for undergrad and get a degree in biochemistry. And then I wanted to study biochemistry more. So that became UCSD in San Diego. And then I started continued homebrewing. And the bottom line is kind of what White Labs San Diego looks like. We're building this one at the top, a couple artist renditions, if you can see these pictures, in Asheville, North Carolina. Anybody been there? A few less people. Asheville is a really small town, so San Diego's over 3 million people in the county, 3.5 million people, but a lot less than Asheville, North Carolina. It's in the western kind of mountains of North Carolina. We went there because a couple of breweries we work with and are big in the industry went there, New Belgium and Sierra Nevada, and some others like Oscar Blue, and this whole homegrown industry like Wicked Weed and others are there. So it's a really active place for craft brewing and home brewing. So we plan to be open there in about September of this year, which will allow us to make a lot more yeast. A lot of the things we've been doing is to try to meet the needs of this craft brewing industry, whether it's home brewers or commercial breweries. And I tell you, it's a challenge. You know, how many hop farmers do you talk to, or if you go to the hop farmers around here, people, you know, you can see they're having a hard time keeping up with the needs of the industry. We have that little brewery in San Diego. We can't get New Zealand hops. You know, I can barely get American hops, and that's true. So all the suppliers are really, struggling to keep up, and some aren't doing it at all, and some are doing it a lot. And I'm trying to do it a lot because I believe that this industry is going to keep growing. So what we kind of do in Westside White Labs is first you got to start with a yeast lab. So this is also kind of like anybody that would grow yeast. This is whether there's another yeast company or another brewery or in home brewing situations, you know, if you're going to do it in your kitchen, this would be the same kind of things you would do. You have to start the yeast somewhere. So we started in the lab. There's a yeast lab in that building that houses all the strains. They've got to be held at minus 80 degrees Celsius, which is pretty cold. That's not a normal freezer, right? Normal freezer, they die much faster than they do in a refrigerator, actually, because of the freeze-thaw cycles mostly, and it's just not cold enough to hold them in that kind of frozen state, too many crystals. So you get into this deep frozen state. It's almost cryogenic type temperatures, and we can store yeast forever. So we create our own yeast bank in there. Wherever we get the strains, we don't keep getting them. We make our own yeast bank, and we keep building and building and building it. When we start yeast from a minus 80 degrees Celsius freeze, so that little 1 mil ampule of yeast is a lifetime supply. Right? We've got lots of backups, we've got different freezers and things like that, but theoretically that'd be about a lifetime supply, because once you have that little 1 mil tube of yeast, you just scrape a few cells off onto a new plate when you want to go back to the freeze. You don't even thaw it out. So really, that looks the same after years and years and years, and it never gets contaminated and things like that. So for us, we have a special team that works with freezers. We have different laminar flow hoods, but we have one just for the freezers. It's our best one, and we really try to put a lot of energy into maintaining these yeasts. Because while our yeast bank might be different from some other supplier, which is fine with me, I mean, our yeast all could be a little unique, they're still supposed to be the same 10 years from now and 20 years from now, and that's our mission, to keep them the same by putting a lot of work into holding the yeast. So we're not just propagating yeast. That's one thing that we do with the cultures we maintain, but we put a lot of effort into maintaining the strains and collecting new strains. Once we get them on Petri dishes, you can kind of see they look something like this. So when you streak it out on a plate like that, you don't see anything for a couple of days, but then the yeast grows up. It's feeding on the sugar, whatever you make in the plate. It could be a wort plate. It could be a nutrient plate of any kind. So sometimes you use plating to look for contaminants by using special plates, or sometimes you use them just to cultivate your yeast on. And you can use slants as well, or sometimes people call them slopes. But it's the same material, just in a test tube that you can screw cap and it doesn't dry out as much. And you slant it like this when it's drying, so it has a big surface area. But a lot of people use the slants to kind of hold yeast for long term. We don't. We use the freezers for long term. They still change on slants. So we mostly work with Petri dishes, and you can really see the yeast a lot better anyway. The whole culture. So anyway, here's when you start a culture. This is the real part of the lab propagation. When you're talking about later, as I'll talk to you about starters and stuff like that, what are you really starting with? How much you're starting with? That's going to determine how much you grow. Well, we start with a few 5 to 10 different colonies and inoculate that into a 5 or 10 mil size right here. And then you just keep stepping it up into higher and higher sizes. This is a 2-liter flask there, and on and on and on and on. And so every couple of days, you move yeast to higher and higher sizes. And ideally, you could keep it going. But if we did that, that's an economical way of doing it, but the yeast would change, right? So we just keep it to a 17-day process, and meanwhile start yeast on plates every day. So we have 10 mil starts, we call them every day, 6 days a week. And then that's making, we got yeast coming off of those systems every day. So when we start packaging the yeast, it's basically once it clears this QC part of the program. So you've got, and this is the same thing again, breweries would do, or you could do at home. You can do different plating when you do yeast. Ideally, every time you do a starter, you should plate it after, right? Or collect yeast from a fermenter, because now you've done something to it. Now, most people aren't going to do that, but that is the only way you really know if you introduced any kind of contamination. So wild yeast plates, general bacteria plates, each one is kind of selected for something different. HLP tubes, looking for lactobacillus and pediococcus, but it takes a lot of effort, and these plates are kind of expensive. But anyway, then we get these pure pitch packages, and like I said, this is just some that I took a picture of. I brought some here, too, so I've got a few, and I can show you those, like I said, but I've got them in the cold room right now. And they've got a manufacturing date, a best before date on there. But anyway, this lot number here, you guys can plug that lot number into this website right there, this QC reports, and it'll give you the QC thing. Because when we ship yeast to commercial breweries, the label's a bit bigger, and it says that QC sheet information on the yeast. So we put that. We make that available for homebrewers. So I think that's kind of unique. There's not too many packages you can pick up in your grocery store, even from big companies, that you can plug a QC number in and you can see the actual lot information. So you can see the plates, their plates on it, and it's got a whole description there, pH, cell count, stuff like that. So you could take that cell count right here and plug into your Mr. Multi or whatever and see exactly how many cells you have in your 20-liter batch. All right, so switching to focus a little bit of the yeast biology. Why are we even talking about yeast right now? What's so significant about them? We know they make the ethanol. Louis Pasteur said in the 1860s that, hey, yeast causes fermentation, and that's great, but brewers knew that, I think, a long time because they were making beer and repitching yeast. But it took a while, I think, for people to realize that, yeah, they make the alcohol, but they also make a lot of the flavor and aroma compounds. And it's kind of, almost something we sort of take for granted. A lot of you know the yeast contribute to the beer, but that's kind of fascinating that they do. It's also something that we as brewers, people before us as brewers, kind of probably help select for to make these yeast do interesting things, because most things that are fermented don't really taste very good. Think of fermented fish or soy or things like that are still specialty foods in lots of different countries, because that's the way historically something could be safe to eat, but they're basically putrefied. They don't taste good, right? But brewers before us have managed to control the yeast fermentation to make it turn into something that's good. And a lot of that is because the yeast has been directed to make awesome flavored aroma compounds. And they do that through their metabolism. So they import these sugars, carbohydrates, glucose, fructose, sucrose, maltose, maltotriose, in that order. Pretty amazing. The yeast do that. But basically, when there's no more glucose is on the receptors on the outside of the cell, because a lot's happening on the cell surface, that's transmitted to the nucleus, and the DNA translation kind of changes to make turn on genes to allow fructose to enter. And then so on to sucrose and maltose, maltotriose. So each carbohydrate you grow the yeast on changes the way the cell behaves. So that's why I think it's important to grow the yeast on maltose, for example, because those maltose genes do certain things from the yeast cell. But regardless, a lot of that is turned into glucose anyway inside the cell, because what is maltose is just two glucoses stuck together. But if you grow it on maltose versus glucose, it still behaves differently. So there's a little bit more to it than just cleaving. But anyway, the yeast take that glucose and they break it down. They're trying to make this ATP right here. That's an energy source. That's the currency yeast use to do things like make proteins and new cells and new membranes. They need ATP. Our cells need ATP. This should sound pretty familiar because yeast are very similar to human cells. Very genetically similar. But they make this ATP, and in normal situations, when there's oxygen around, yeast love oxygen, our cells love oxygen. It takes pyruvate here, which is kind of cut down from glucose. A little bit of ATP is made. And then normally you have oxygen in the Krebs cycle of the mitochondria. You make a whole bunch more ATP, and everybody's happy. About 36, 38 ATPs from each glucose. Well, we aren't giving our fermentations oxygen, right? We do that right from the beginning. You were told that right from the beginning. Hey, don't give your fermentation oxygen because the beer's not going to taste very good. And even if you did, there's so much carbohydrate there, the yeast is not going to really do this respiration with it anyway. What they are going to do is they want to continue to make ATP, so they just turn this pyruvate into ethanol real quick, and the ethanol is pushed outside of the cell, or goes outside the cell, so they can make this cofactor NAD, so it's used here to make ATP. But they don't get 36 to 38, they only get 2, a net of 2. So that explains this low little bit of energy that yeast get in an anaerobic fermentation, explains why they're a little fragile afterwards, right? They don't last very long when you collect it. They didn't make a lot of energy in this anaerobic fermentation. They might have made a great beer, and we slowed them down in all sorts of ways, but they didn't make a lot of energy. So that ethanol, along with CO2, because as we make ethanol, we make CO2, that's pretty cool. So we can carbonate our beer with the very byproduct of the yeast fermentation. Even though a lot of people add CO2 now, we didn't do that until recent times, right? We captured that gas inside the beer, and we had a nice bubbly thing with ethanol in it. But you can imagine all these little steps. This is not one step. Each carbon bond that's broken is a different enzyme, and so you got to keep breaking bonds, and you make all these different substrates in there for the next step that then maybe go to the next step, or maybe goes to an amino acid production step, or something like that. And so these things never really go to 100%, so you're making all these different carbon skeletons that can turn into esters and fusyl alcohols and other things. And so that's where we generate a lot of those flavor and aroma compounds, is through the byproducts of these various steps of metabolism. And those can come from, this carbohydrate metabolism. It can come from the fusyl, the amino acid metabolism over here is what I'm trying to show with nitrogen. And then fatty acid metabolism over here. So some of that pyruvate does go over here and with some oxygen, and we make our unsaturated fatty acids and our sterols. Those are going to be really important in the membranes as we make new cells, and with oxygen, which is going to be a big part of what I'm going to talk about. So these three kind of parts of metabolism, carbohydrate metabolism, fatty acid metabolism, and amino acid metabolism, those three main pathways kind of going on is what generates a lot of the flavor and aroma compounds. Then, though I talked about it because I think we all should know about it, you actually don't have a lot of control over it, right? You put the yeast in and it does its thing. It does this stuff. It's not like you can say, do this and this and this, but you can a little bit the more you learn about it through temperature control, through how much oxygen you give yeast and stuff like that. So you have some controls on these things, but for most part, yeast is trying to consume carbohydrates and grow. And we're just doing it in a way that we get what we want. And so each yeast strain makes different flavors. This picture is just to kind of show you that you can split wort into different fermenters. Like I said, we do that on a big scale. We're taking 24 hectoliter batches, splitting it into 5s. You can do that on a small scale. You can do that in liter bottles. A lot of homebrewers, you've probably done, because you can put them in that 20-liter batch in a 10-liter fermenters, or 40 in the 20s, and pitch different yeast. And how many people have done that here? Yeah, a bunch of you, right? That's one thing we're supposed to be doing in homebrewing, right? Is probably learning something from every batch, and that's a way you can really go, those yeasts have a big influence on beer. And they look different, they'll behave different, but they also make different flavor aroma compounds because there's small differences in each strain's DNA that's going to change one of those enzyme steps, or how much of that particular enzyme is made, right? Because we have multiple copies of each chromosome in brewer's yeast, so the copy number can have an influence, and each strain has a different copy number sometimes. So there's a lot of things going on, and then the regulation of that, it makes each strain different. If you look around here, we're all Homo sapiens, but we're all a little different. We're all individual strains, if you will, right? And we know that because we can look at each other and know that. We don't have the ability of cloning ourselves and keeping us going, but that's what brewers yeast do. So a whole room of different strains, maybe. Like we said, we make about 100 different strains a week, and there's a lot more than that out there. But the brewer's yeast has this cool property of cloning itself. It's an asexual form of reproduction. So barring any kind of mutations, as we try to keep them low by keeping yeast healthy and happy, it's going to stay the same. Now, yeast out in the wild, they mate, but brewer's yeast has lost that ability to mate because it doesn't have to. It's in this nutrient-rich wort with plenty of carbohydrates, which is pretty hard to find out in the wild, right? Except at certain times, and ripe fruit and stuff. But so they become really unique in our hands. Here's an example with 7 different yeast strains there. California ale yeast, Irish ale yeast 004, European, these are white Labs yeast, Burton ale yeast, California 5, Hefeweizen, and Belgian Golden, where we did a fermentation with the same wort, split it into these different fermentations, and then measured the esters and fusel alcohols that came from it. And we have all this data online too, and I have lots of different slides of it. I just wanted to show you one that showed you the fusel alcohols on the top and some esters on the bottom. And out of 30 or 40 of these, usually you're measuring 2 or 3 and saying there's a total representation of the esters or total representation of fusel alcohols. You can't really do this at home probably because you have to get the beer in a gas phase. Right? So a gas chromatograph is what we use in the lab to volatilize the beer to the gas phase and then measure the retention time of these different compounds. But we can do that and measure that. So 1-propanol and isolated alcohol, these are 2 fusel alcohols, and you can see the flavor thresholds are different. So you need a lot of 1-propanol, 600 to 800 ppm to taste it. So you're generally not tasting that beer, but the level of it gives you an indication of how much fusel alcohol is there. And California ale yeast has 37, California 5 is 20, and then the Hefeweizen is 30. You can see this Belgian Golden yeast is 80. So the Belgian yeast tend to make more fusel alcohols. The unique thing about that is the fusel alcohols are what turn into esters when it combines with an acid. So first that cell, after about 12 hours into the fermentation, starts making the fusel alcohols, and that's where the esters come from. So if you're going to make more fusel alcohols here, these Belgian yeast, you're probably going to make more of the esters. And here's some esters below here, ethyl acetate and isolated malacetate, and they have some different flavor thresholds. So ethyl acetate is not a real pleasant ester. People describe that as nail varnish remover, nail polish remover. So I think you might be able to think about how that smells. Not particularly good in beer, but you need a lot of it to smell like that, right? But it's a good also example in brewing how there's good things and bad things that come from our fermentation, so we're kind of trying to control the good things. Isolated acetate is one that a lot of us like in certain beers because that's the banana ester. You'd like that at Hefeweizen usually, right? Probably not in a pale ale. So you want to use a yeast that's going to be appropriate for that. And there's fermentation conditions you can change and stuff like that, but the point of this is it's embedded in the genetics of these different strains. So you only have so much control over it, because I can show you right here, California ale yeast in this fermentation produced 17 ppm of, 0.5 of isoamyl acetate. That's below the 1 to 2 threshold. So you use California ale yeast in an IPA or pale ale or even a wheat beer, it's not going to have that banana ester, no matter what you do to it. Irish, a little bit more, but still in the threshold. European, a little bit more on the threshold, and so on down the list. But look, we get to the Hefeweizen yeast that we sell the most of, this WLP 300, and it's 12.8, almost 13 ppm isoamyl acetate, so quite a bit more than that 1 or 2, right? So that is why you use that yeast in a wheat beer, and you get that banana ester. So you can do different things with malt, you can do different things people talk about, but really it's the yeast strain that's going to have the most influence on how much of that isoamine acetate is produced. And that Belgian golden yeast that makes lots of esters, that had lots of fusel alcohols, it's still almost half of what the Hefeweizen makes in that banana ester. So if you use that Belgian strain in a wheat beer recipe, it's going to have all these Belgian-like characteristics to you with the phenols and stuff, but it's going to be missing some of that banana ester component. That's where it comes back to strain selection. And these strains are different, like I said, mostly because of the DNA inside those yeast strains. So we started this study a few years ago. We were fortunate to have a local company in San Diego called Illumina, and they make these high-throughput DNA sequencing machines. Very, very expensive, but they like beer and they're interested in this study, so they're helping us with this. Because I can't buy a $10 million machine. $50,000 is about right for a machine for me. But we were able to sequence 96 of our yeast strains, and then through another 100. And it's become a big collaboration, and mostly with this lab in Belgium, besides the people in San Diego that are helping. And this is unique, really, because this is the first time that brewer's yeast has had its full genome sequenced. Saccharomyces cerevisiae lab strains was a long time ago. That began the Human Genome Project, but that wasn't brewer's yeast. So brewer's yeast, even though Saccharomyces cerevisiae are quite different, they have a lot more DNA than lab strains, and that's one reason why they weren't sequenced before. And that's also why we still don't know why brewer's yeast does what it does. We know that can say, hey, Hefeweizen makes more of that banana ester. Well, why? And what other strain might that be more related to? So that's what we can start figuring out as we look at the full DNA complement of brewer's yeast. Just like a lot of other organisms now with this technology, we've been able to look at their full DNA and go, oh, wow, here's a whole bunch of things about this organism we didn't even know about, or our own human evolution. So you're going to see this publication come out in the summer. But just as a little bit of example, we're able to get yeast in these different groups. This is kind of a phylogenetic tree, where it's kind of what they do combined with the genetics of the yeast strains. So we found that there's a beer group 1, and then another beer group 2, an Asian group of yeast that are different genetically than these beer group 1. So the interesting thing about that, there's a whole bunch here, but just to kind of whet your appetite is, this beer group 2 that are more similar to these wine strains, the C, are also the high alcohol strains. So, and wine strains generally are high alcohol tolerant. And so we think, how can we get our yeast strains sometimes more alcohol tolerant? Well, you have some ways maybe, but you also need to use the right strain from a genetic aspect. Some are going to be more high alcohol tolerant. So once we have this information, I think it's going to make even strain selection more interesting and for you, because you're going to be able to have this kind of genetic information to make yeast choices. So that'll be coming down the pipe, and this just kind of shows that ethanol tolerance, I said, with wine yeast. You've got the sake yeast up here, which is part of that Asian group, that also are alcohol tolerant. And then this is just kind of going through some differences with liquid and dry yeast, because a lot of people ask me that because we make liquid yeast. If these membranes are going to be really important with yeast, right? That's where all that trafficking happens. That's where flocculation happens on the cell wall on the outside. So if you strip most of that water out, and you can look at electron micrographs of yeast this way, they're pretty shrinkled up. So you rehydrate them, and you should. I know a lot of people don't when you're using dry yeast, and often even not told to in the instructions. But it makes a big difference on how they're going to ferment, especially when it comes to, say, something smaller scale like bottle conditioning. We've done a lot of work on that as well. And you don't have to have successful fermentation, so that kind of thing continues on, but it makes a big difference in how they ferment. And the eventual flavors that they produce, and the flocculation, and the attenuation, these properties of yeast cells that are really important in fermentation. So there's some cell death, there's cell wall changes, plasma membrane changes. And then when they're rehydrated, because we make some dry yeast as well, just on the experimental side, they don't export those esters and things the same. So the aromas are a little bit different. Comments of what's different about liquid yeast and some myths. I've got to speed up a little bit here. Just some comments. Traditional strains, so the tradition of our industry was liquid because that's what brewers did. They repitched and repitched and repitched. So that's how we've ended up with the yeast strains that we have today. So those strains that make the Belgian styles, and make the wheat beers and make the other things, but also all of the ale yeast that were used in England kind of developed in this liquid format. How do you handle yeast? When you're using yeast in your brewery, it's in liquid form. The biggest breweries, most of the biggest breweries in the world, use yeast in liquid form as well. And so they're kind of propagating it for themselves. But whether you're propagating it or you're now using it in your brewery, you start with pitching it into your beer, collecting it, storing it, repitching, and how many times, how many strains can you use. So this is where it kind of starts bordering on commercial breweries a little bit, because they do repitch. That's, again, how breweries have done it forever. And they used to repitch forever, but now that you don't have to, a lot of them will just go about 10 generations. Home brewers, a lot of you probably are also repitching a little bit, but generally it's more like a couple, 2 or 3 times, because the yeast is sitting for a longer period of time between each repitching, and it loses some viability. After about a month, yeast in your refrigerator, yeast that's collected from your fermenter, from the moment that the fermentation's done, after about 30 days, it's 50% dead. Or you can look at it as 50% alive. So that's why you'll hear kind of that number of about 2 weeks. If you use yeast within 2 weeks at the end of fermentation, you're going to be pretty safe. But once it's a month old, it's 50% dead. And that's because it was an anaerobic fermentation. You only make 2 ATPs at a time. You're not going to have a lot of energy stored there for survival once you collect it. So that's why you want to get it going again. Yeast are quite happy to keep fermenting. Collect it, reuse it. Collect it, reuse it. And that's what's going to be done in commercial breweries. But again, in home brewing, it's not always that way, because we want to make a different style of beer. And we want to use a different yeast strain. How much are you going to use? Here's some examples of dry yeast, liquid yeast concentration. So liquid yeast is generally around, it's something like this, or when you collect it from your beer, around 1 to 3 billion cells per mil, 10 to the 9th. So they're very small. So we're talking about big numbers. And then when you dry it, you go from that 1 to 3 billion to 10 to 20 billion. Now it's per gram, but gram per mil is about the same. Yeast is it's pretty close to the density of water. Yeast cells are mostly water. So you can see you get this big jump in concentration. Baker's yeast tend to be closer to that 20 billion per gram. Brewer's yeast close to that 10 billion per gram, because brewer's yeast is much bigger. Right? True brewer's yeast strains are a lot bigger than Baker's yeast or other Saccharomyces cerevisiae. Beer inoculated with yeast tends to be 5 to 10 million cells per mil, or 15, and then it grows to about 25 to 50 to 70 million cells per mil. So that growth is going to be key, right? When you're talking about managing your fermentation, it's not yeast is just consuming sugars and making these flavor compounds. It's growing. So the better you control that growth phase, from how much you add to how much oxygen you give it, what temperature you keep it at, is going to help determine that growth is the same every time, and you're making the same flavors every time, because those flavors are coming through that growth. So just an example, if you were going to inoculate 20 liters of beer and you wanted 5 to 10 million cells per mil, knowing these things are how you know how much yeast to add. So if you want to play with the numbers and get really familiar with the numbers, that tells you a lot of things. 10 grams of 20 liters would give you, if it's 10 billion per mil, would give you 5 million cells per mil. If you added 20 grams in 20 liters, you have 10 billion cells per mil here. If you were at 10 billion per gram, and that's at 100% viability too. Liquid yeast at 3 billion per mil. If you use 33 mils in 20 liters, that's 5. 66 in 20 liters, that'd be 10 million cells per mil. So you can see, knowing these numbers helps you kind of judge how much you need of each amount. It's not like you can achieve the same things with different amounts of yeast, even if it's coming from your beer, but that's going to be in the millions of cells per mil if it's not slurry. So getting good with the calculations can help you exactly determine how much yeast you need. I mean, you can go on Mr. Malty, like I'll talk about in a second, and that's awesome, but that's an estimation, right? I mean, that's not your sample. If you want to pitch yeast by gravity, that's also something brewers realized a long time ago, that the higher the gravity of the beer, the more yeast you add, right? So the million cells per mil per degree Plato is this per degree Plato is in there because brewers realized, hey, if I make a bigger beer, I need more yeast. That's not on this label right now, but we've got a new label coming out that has this whole chart on the back of it. And those labels are going to start going on those next month. But here's 20 liters, 40, and 60, up to 12 Plato, so 1048 is using one package of pure pitch package and no starter, up to 3 for 60 liters, and different amounts for different gravities. And also putting starters on here. So I'm trying to combine the information that's different than it was 20 years ago with how some homebrewers are handling yeast now, and either giving some starter recommendations or multiple package recommendations for high gravity beers. You can always use one of these, or one dry yeast, or one Y yeast, or whatever, for 20 liters of beer, and they'll work. That's the way it was designed. But if you want to make a big IPA and you want to use more yeast, this gives you the examples to do it, or a chart to do it. And there's also, like I said, different ones online. Jamil Zanishev's Mr. Malty, he co-wrote the yeast book with me, and he's got a website that has a chart like this on there, and a lot of people are using that to determine their starter size or how many packages they buy. The different thing here is he's got a calculation that gives you the viability percentage. Now, you can always unclick that, put it on a microscope, and count your own viability. Then you'll really know how much to use. But if it's an estimate, this is an estimate from one experiment done a few years ago, which is based on experiment, which is good. But if you put a yeast you buy from White Labs, you put a date in there, it's going to generally tell you a viability that's way below what it really is. So in some ways, that's good insurance. This just tells you to make a bigger starter, not that big a deal. But you don't always have to. If you know the exact viability, you'll see that if you punch that in, you could actually make quite a smaller size starter. But whatever size you make, I do like the stir plate. It's not necessarily about adding oxygen, but it does keep the yeast in suspension well and keeps them growing. You get a few more cells out of it. It does not produce a good amount of storage, carbohydrates in the yeast, the glycogen, so they won't store very well, say if you make a starter this way. But most people aren't doing that. You're just making a starter and you're using it. But I wouldn't make a starter like this and then wait a week before you use it. You can see this one doesn't have an airlock on it. You don't really need to. A nice piece of foil and a foam stopper is how Labs do it. But there's nothing really wrong with an airlock. So starters are, how many people here use starters? Yeah, a lot of people. That was about maybe half the people. And they're one of those things I always tell people, if you like making starters, make starters. But if you don't like to, you don't have to either. You can use a chart like the one I showed you before to figure out using one package or how many packages you want to use, so yeast you buy. And then regardless if you're using a starter or you're just starting with yeast, the fermentations kind of look like this. The first few hours, yeast uses up the dissolved oxygen. And it's not like it uses the oxygen and then you see fermentation start. It takes up the oxygen. It's going to use that oxygen to make plasma membrane materials. That was maybe on a timer there. But anyway, we'll get right into that oxygen because I'm getting a little bit long here. So what are yeast going to do with this oxygen? They're going to make new yeast cells. So remember, yeast have to grow in the fermentation. So if one cell is going to grow into 5 cells, it needs more membrane materials. The only way you can make that membrane materials is with the unsaturated fatty acids and the sterols that you're going to get from adding oxygen, or some people add olive oil to get some of those fatty acids. But regardless, if you don't add the olive oil, you need some way of getting in more membrane materials. So that's what the oxygen is for. And most of you are not aerating right. I got to kind of say that, because if I talk about oxygen, a lot of people go, oh yeah, it's pretty good. But it's really not. Even half the breweries that we measure the dissolved oxygen in, who feel that they've got it all dialed in because it's in line, are at half the recommended level. So as homebrewers, what chance do we stand, right, of getting to the full level? But we actually can, because we're dealing with smaller volumes. So 1 or 2 minutes of pure oxygen from a bottle like that into a batch of 20 liters gets you usually between 8 to 10 ppm dissolved oxygen. Sometimes those pure oxygen bottles are hard to get, or we don't want to have some more equipment in our brewery. Another way to do it excuse me, would be that kind of fish aeration pump. Just a little pump that sucks in air from the environment and pushes it into an aquarium, because fish need oxygen as well. You can use that to aerate your wort, but it's air, not pure oxygen, so you need longer contact times, and it never really gets to the 8 to 10 ppm, but it holds it at 3 or 4 a little longer. So if you do that for 30 to 60 minutes, you can also get the right amount of dissolved oxygen. But if you want to shake, you're never going to have the right dissolved oxygen, and you will always have some weird fermentations. It will look, it will look okay because it ferments, but it's not going to ferment as far as it should. It's going to be sweeter than it should, and it's going to have some different characteristics. Aeration is just as important as how much yeast you add. So a lot of times we're spending time thinking about how much yeast we should add. We're going to Mr. Multi, we're doing that stuff, and we're doing starters, and then we're ignoring the oxygen part. They go hand in hand. Adding more oxygen is the same as adding more yeast, because that's how you get more yeast once you get into fermentation. Because no matter how much you add in the beginning, you have to grow more in the fermentation, and that's where the oxygen comes in. So everybody can improve their home brewing by getting in the perfect amount of dissolved oxygen. It'll take a whole few points of gravity off the beer, and even those commercial breweries I said that think it's perfect because it's foaming doesn't mean that's what they're seeing. They're seeing foaming in line. That doesn't mean that oxygen is solubilizing, getting into the beer, into the wort. So this kind of shows you that some numbers here. Shaking for 5 minutes, 2 ppm, 3 ppm. So that's 20 or 30% of how much oxygen you need. So fermentation is going to work, but you're going to get stuck, or they're not going to finish. And 30 seconds of pure oxygen, 60 seconds, 120 seconds. So there's not much difference between 1 and 2 minutes. You're at 9 to 14 here. But just a little graph we did a few years ago. I just want to show you how that affects fermentation rate. So here's the cell count. There's a 19, a 10, and a 5 ppm dissolved oxygen here. And it's kind of hard to see this pointer, but it's impossible to see this pointer. But the line. So here's the cell count going up. They're both pitched at the same level, all 3 of those rates, but the cells grow in the fermentation. So you can see that the 10 and the 20 ppm grow more cells than the 1 and 5. That's the yellow line that stays low, right there. So what does that mean if the cells don't grow the same or as much as they should? They go slower in the fermentation. So here's the yellow line again, showing the fermentation rate, and the specific gravity is lower on the 2 with higher dissolved oxygen. And that trails out through the whole fermentation. And kind of it matches at the end here at 168 hours. But sometimes that one that didn't have as much dissolved oxygen never reaches the same final gravity. So the difference between 2 Plato and 4 Plato or 3 Plato is a pretty big difference. Less alcohol, more sweet, and you're kind of leaving part of the beer unfermented. So dissolved oxygen is going to be really, really critical. And I'd also say temperature control, just didn't have time to cover. is also really important, right? But that's about the flavor development. Okay? That's kind of controlling the flavor development. But that's also something we think about sometimes, that the temperature control, the aeration, and how much yeast you add. Those are some of the controls you really have. And then what else you can do is use different yeast strains, right? So that's where it gets kind of limited to your imagination. We make over 100 different strains. You can also blend different yeast strains together, or you can use wild things like this. So that's a bacteria that a little close-up looks a little weird. But red bacterial fermentations, Brettanomyces is not a bacteria, it's a wild yeast. And it's in the air. It's much more prevalent in the air than Saccharomyces is. So it used to contaminate beer all the time. Louis Pasteur said, hey, yeast is doing the fermentation, but these wild yeast and this bacteria is causing our off flavors. And over the next 100 years, after the 1860s, brewers started cleaning. It took a few generations. A new brewer really has to come in and say, I'm going to change something. So they started cleaning, and now the Brettanomyces and the bacteria are out of most of our breweries, out of most of our wineries. But as I said yesterday to some of the home brewers there, you're adding it back in on purpose sometimes now. So we're adding Brettanomyces because some of us like that characteristic. We're adding bacteria like Lactobacillus, Pediococcus, and Acetobacter. Well, usually not Acetobacter, but that could get in there if you're doing a native fermentation, an unpitched fermentation. That's going to get in there. Here's what a wild fermentation system might look like. This is a cool ship from Cantillon. You can see this is on the top floor of that brewery, and there's a lot of these still in Belgium. Even if they're not doing wild fermentation sour beers, they might still cool beer at once like this. Anyway, so that hot wort is put on into a thin layer onto a big pan, so it cools overnight. But as it's cooling, it's going to pull in a lot of Brettanomyces and bacteria from there, and hopefully some Saccharomyces that might be in the wood and stuff up here. You can see some openings there. Used to be even more open before EU putting different kind of restrictions on what breweries should do. But they'll leave it in there, and then they'll put it in barrels and ferment it in barrels. So other people that used to do this, because you still have to cool your wort somehow, 100 years ago, they know it would get sour if they left it there, so they might cool it down a little bit and then move it into something else. They wouldn't leave it for long periods of time open like this without pitching it. There's also kettle souring. Anybody doing any kettle souring? Yeah, it's become kind of a big deal with home brewers and some commercial breweries because it's sort of a cleaner way of doing sour beers because you're going to boil it again after. So you mash as normal, you lauter as normal, then you either don't boil or do a quick boil. I think most people are leaning to this quick boil because if you don't, there's lots of enterobacteria and other things that will grow along with this souring process that get quite foul. And a lot of times, you haven't added your hops yet because you're not doing your boil. Then you cool it, you add lactobacillus. We make those cultures. Some people are culturing up some yogurt and adding lactobacillus that way, waiting for the pH to drop to about 3.4, between 3.4 and in the higher 3s. But some people don't like it. And this is a shirt I found online. Got some cattle sours. And I think the thinking is they're like, oh, you want an easy way to make sour beers? Well, then don't do it, And so sour beers are supposed to be difficult, artisanal, something that you toy with, and not you just don't do it in your mash tun and then you've got a sour beer. I don't really hold that theory. I think it's funny and I think it's interesting, and I know there's different sides to it. But the one thing that is very true is kettle sours are becoming more popular. Do they have the complexity of a 3-year-old sour beer in barrels and stuff? No, they don't. But they have some sourness, and that was biologically derived. It wasn't just somebody adding lactic acid in. So I think we got to keep that in mind with all of our beer fermentations. That we're already developing all these flavors and characteristics biologically. Let's keep that going that way. Or do you want to culture your own yeast? This is a picture for Rogue Brewery, and the brewer there, John Mayer, wanted to retire, or has been talking about retirement. I don't think he's retired yet. Anyway, they wanted a yeast collection from kind of that Newport, Oregon area. So we were employed to help them out with that, and they wrote about that, so it's not a secret. We went up there and we did this yeast collection, and one of the brewers thought it was a joke to just swab John Mayer's beer. We took that back to San Diego, and that yeast swab from his beard produced the best yeast out of that collection. So they made a beer with it, wrote Beard Beer. I get asked about this quite a bit too. This got a lot of attention at the time from the New York Times and other newspapers, and you think it's sort of a gimmick, but it wasn't. It was a true lab experiment that produced something that they said, hey, let's run with this and let's make a beer with this. Because once you purified that yeast, it's not really in the beard anymore, but it did start there. And it's not their brewer's yeast, and it's not any of the other yeast they had in the brewery. But it's kind of Belgian-like, the beer, because any wild yeast is going to have that kind of Belgian-like character, which is phenols, really. Just the last slide here to show you that this is something we put online to kind of increase the number of strains like that you might want. It's called the Vault. So it's on whitelabs.com, but then there's this Vault page. You guys can go there as homebrewers and vote on different strains that we should make. And you vote with your credit card. You're basically pre-ordering it. We charge that credit card when we start making the yeast, but that allows us to make a production run and get some of these unique strains that we have deep frozen away, and that's why we call it the Vault, to make for you. So that was launched just a few months ago. We've already made a couple of strains. We've only released those in the United States because we're direct shipping those to the home brewers. But the idea is that'll trigger a bigger batch that we can make, and then a store here, for example, can say, I want so many, and then those strains will come here. So you'll see that grow and proliferate as we figure out how to integrate that into our different systems. But this is one new way we have kind of devised to get some new strains out there beyond the 100 or so we already make every week right now. And with that, I'd like to say thank you, and maybe I have time for a few questions, maybe not. Okay, thank you.
Well, thanks again to Dr. Chris White for letting me record and air this keynote. Be sure to go back to July 23rd of this past year in the archives and listen to our one-on-one conversation across the conference table, up there or down or over there, whichever direction you are relative to San Diego. But we went to White Labs and talked to Chris right there, face to face. Listen to that if you haven't. I may have to dig out my aeration stone and go back to the hardware store for a fresh tank of oxygen. I'm a little ashamed. I get pretty good beers by shaking, but maybe they could even be better if I'm a little less lazy. In the meantime, if you have brewing questions, show suggestions, or just want to say howdy, write to james@basicbrewing.com, or just fill out the contact form at basicbrewing.com, and please don't forget to tell us where you're from. Check out our support link, where you can throw a couple of bucks into the tip jar by subscribing financially to our podcasts. Be sure to check out our DVDs, Extract Brewing and Partial Mashing, Stepping into All Grain, Low-Tech Lagering and Decoction Mashing, and Introduction to Wine Kits. You can find them all on our site. Get a free Basic Brewing bottle opener with any DVD combo. You can check out our Basic Brewing shirts in the store as well. You can find our log books, where you can track and log up to 50 batches of beer. Check out all those at basicbrewingshop.com. Thanks to everybody who's continued to click on our Amazon.com link. We appreciate the support there. Our featured products this week that were purchased through the link are the Saggy Baggy Elephant, Step into Reading, and Black Cherry Tomato Seeds, Rare Russian Variety, 30-plus seeds. Love them cherry tomatoes. Thanks again, everybody, and remember, I can't tell who bought what, so no worries there. Just click on the Amazon.com logo on our site the next time you feel like Amazon shopping. We greatly appreciate your support. And don't forget, you can also join the American Homebrewers Association and subscribe to Brew Your Own magazine through the associate links on basicbrewing.com. That's all until next time. Until then, thanks for listening, everybody. I'm James Spencer. Production help for Basic Brewing Radio and our website is provided by Kelly Dodson. Basic Brewing Radio is a production of Active Voicing. We'll talk to you next time, everybody. So long.